Hello everyone, welcome to section 2. This is one of my favorites. In this one, well, I have a few favorites, but this is one of them. We're going to be covering in this first section neurons and neuron structure, action potentials. We'll talk about neurotransmission and neurotransmitters. Then we'll get into the nervous system. We'll talk about the brain and all the cool things going on there. And then the second half of this section is endocrinology and hormones. So a lot of what we'll talk about here is communication in the body. So how neurotransmitters and hormones carry information throughout the body. So let's start with, what is a neuron? What do they look like? What a neuron is, it's a cell that carries information. And there's about 88 billion of them, and they've just changed that. It was a big thing in science. They used to say it was 100 billion, but they've just re-estimated. Now it's only 88 billion. But these 88 billion neurons have 100 approximately trillion synaptic connections. That means thousands of them interact and connect with each other and forms this really dense web of connections. And 100 trillion synaptic connections, that's more than the stars in the galaxy. Oh, there's a lot going on in your brain. Let's see. They're pretty narrow. They're only about 0.01 to 0.05 millimeters in diameter, a neuron. And so they're very thin in humans. There is a species, the giant squid axon. You can see a giant squid axon with your naked eye. They're about a millimeter. You can see them in the naked eye. And so we study those. But there's not a lot of neurons in a squid. They don't have 88 billion because they're just not as cognitively dense, and there's not as much going on there. And so our neurons, if they were that wide, they wouldn't fit in the room, basically. So ours are much more narrow, and there's other things we do to speed up transmission. Let's see, what else? We talk about nerve tissue, and you think of neurons. Neurons get all the press, they get all the excitement. But there's another type of nervous tissue called neuroglia. And there's a lot more neuroglia than there are neurons. And back in the day, when I was in grad school in the 90s, and my PhD's in neuroscience, we got maybe an hour on neuroglia. It really wasn't thought to be that important. Neuroglia means nerve glue, and it was thought that these cells just supported the neurons. But we got 10 to 50 times as many neuroglial cells as we do neurons. You don't take up that much space. You don't have that many cells if they're just supporting the rock stars, the neurons. And it turns out they have such important functions. And if you take neuroscience with me, we'll spend more than an hour talking about neuroglia. They're very important. So maybe you've heard of astrocytes. We'll mention Schwann cells, oligodendrocytes, but they have many, many important functions. Now your brain itself, it's only about 3 pounds, and it's gush, as we'll talk about later. It's mushy. It really doesn't even support its own weight very well. It's kind of texture of soft tofu, but that brain contains multitudes. So let's look at the anatomy of a neuron. Form follows function. These cells, many of them are very, very quite small, but some of them, there are some as long as a meter or more, and they look kind of like a wire. The main wiring part is called the axon. And the axon is this long wire, and form follows function, meaning what their function is to pick up signals. So there's a lot of receptive surface area here, pick up signals and carry a signal throughout the body. Now they may only carry it a millimeter or even less, or they may carry it a meter or even a little bit more, but this is going to be the axon that carries information. So let's label the neuron, and I will want you to know these different parts. Dendrites, we'll talk about all of them. Dendrites, that's the main receptive surface that picks up the signal. So these little branches. Now when you're looking at a neuron, don't get confused with these branchy things and these branchy things over here. So look for the cell body, okay? This one is called the axon terminal. These are the dendrites. What I do is, look at your hand, stick out your arm, and your palm would be the cell body. Now wiggle your fingers. So this would be your palm. Here's your fingers coming off of the palm, off the cell body. Here's your arm is the axon. You can get a sense of that. Okay, so dendrites, axon terminal. The axon itself is the wire. Myelin. Myelin is this fatty substance that covers axon. Not every axon, not every neuron in your body is myelinated, but the ones that you want a fast signal for, those are myelinated. We'll talk about those in just a bit, but those are going to speed the signal and make it less likely to, it makes it go more efficiently. And you do have to leave little naked spots, which we'll talk about. So here is your cell body or soma, and the nucleus, which is going to create new proteins. This is the axon. This is the myelin. The naked bits of axon in between the myelin are called node of Ranvier. They're nodes of Ranvier. And what we're going to see is how signals travel down an axon, down a neuron, is it's a change in voltage. It's just going to be the movement of sodium and potassium ions. So sodium is going to rush in, potassium rush out. Sodium rushes in, potassium rushes out. And that's going to happen at the nodes. And it just makes the signal go much faster. Because if you don't want to have sodium rushing in here, and here, and here, and here, and here, and here, and here. No, here, to there, to there, to there. So these nodes of Ranvier, there's a lot of sodium channels. This will all make more sense in a couple of videos. This is just an overview, so don't panic yet. But node of Ranvier, our action potential, our signal is going to hop from node to node to node and carry it down the axon that way. Neuroglia, we just talked about those. There's a lot of different types of neuroglia, and two types of neuroglia, oligodendrocytes and Schwann cells, make the myelin. Oligodendrocytes make it in the central nervous system, and Schwann cells make it in the peripheral nervous system. So these neuroglia cells, those are the cells that make the myelin. Okay, so all we've done so far is just label this. Don't panic if you shouldn't be on page 4 or anything yet. We're still halfway through, right at the beginning of page 1. So let's look in more detail about all of this. So when I throw a neuron in your face, there's wiggly bits here, there's branchy bits here, and branchy bits here. Dendrites are going to be the ones that are attached to the cell body. So it's like the fingers attached to your palm. So the soma, or cell body, that's going to carry out the basic processes of the cell to keep the cell alive. So that's where the nucleus is. It's going to make more proteins. That's where Golgi bodies and Golgi apparatus and mitochondria and ribosomes and all of those are in the cell body. Keep the cell alive. All right. Dendrites. Dendrites are your main receptive surface of the neuron. What that means is they receive the signal. There's going to be another neuron here that's going to spit a neurotransmitter. Okay, neurotransmitters are released from the axon terminal. And imagine there's another cell here with the dendrites right here. And so you're spitting neurotransmitter onto your dendrites, and your dendrites are going to pick up that signal. So they're the main receptive surface. They pick up the signal that was released from another neuron. And then they've picked up the signal, and it actually is going to bind to something called receptors on the dendrites. It's going to pick up those signals, and if you get enough of it, it's going to send the signal to the cell body, and then it's going to go to a part called the axon the axon hillock, which is right here. We'll talk about that. So these dendrites pick up signals, and they're going to send them through the cell body to the axon hillock. So what these dendrites are, they're branches. Branched outgrowths that come from off of the cell body. And when we draw this in class, we always draw one-to-one, one neuron affecting another neuron. But in your body, that's not how it is. It can be thousands. It can be as many, it can be 10,000 individual neurons affecting one other neuron. And then that one other neuron has branches at the axon terminal, so it can affect hundreds usually, or up to 1,000 others. So that's how we get these 100 trillion synaptic connections. It's not one-to-one. There's thousands of one neuron affecting another neuron. And we call that the dendritic tree. So It's connected to thousands of neighboring neurons. And this neuron might say, go. And this neuron might say, stop. And this one says, stop. And this one says, stop. So it's going to kind of add up all the information and make a decision what to do based on, which one wins. Now on the dendrites, there are these branches, like wiggle your fingers, and they look like branches, but the branches have leaves on them basically. And they're called dendritic spines. And that's to increase surface area. And that's to receive the signal. These dendritic spines have little proteins on them, little receptor proteins. And these dendritic spines increase and decrease. Basically, use it or lose it. And there have been studies, some of them sad, but basically the increase, the more you use it, the more it grows, the more synaptic connections you can have. It deepens and enriches that synaptic web. If you don't use a connection, it goes away. So one example, they did studies that they would stimulate a monkey's palm, and they'd rub and rub and rub their left palm. And then they, sacrificed the monkey and dissected its brain. These were done decades and decades ago. And the area of the brain that corresponded to touch from the left palm had more proteins, more connections. Now, another sad study, but taught us things about the brain, this was done in the 50s. They took newborn kittens, normal newborn healthy kittens, and blindfolded them. And then just took the blindfold off after a couple of weeks. And then those kittens were blind for the rest of their life because they did not use these connections that at a very important time of development, when you're a fetus, when you're an infant, that's some really important time of brain development. They didn't use them for those weeks, and they lost the ability to have those connections. So these senses, these dendritic spines can increase and decrease depending on use. That's why you practice something, it gets better. All right, next is the axon. Axon is the long wire, and its main function is to carry the signal. It's going to carry the impulse away from the cell body, only away from the cell body, to the axon terminal. So remember, the dendrites are picking up a signal, and we'll talk about what that signal is and how it's carried. We'll get to that. But it's picking up a signal, sends it here, and once it gets the axon hillock, the beginning of the axon, it goes quickly down the axon. Right? And then it's going to cause the axon terminal to release neurotransmitter. Now, the way axons carry information, it's called an action potential. And we'll talk about that in later videos. Axons vary greatly in size. Some are less than a millimeter. They can be a few hundred micrometers long, and some can be longer than a millimeter. Most are a few millimeters long. It depends on where they are, where they're carrying information to. The very beginning of the axon is called the axon hillock. That's where it meets the soma, and that's where you start an action potential. The axon terminal, remember there's going to be a lot more branches than this, that is where you release the neurotransmitter. And every neuron only has one axon, but sometimes there can be a little, what we call a collateral, a little thing coming off here. It's not considered the main axon, but it's actually a little branch, and it can feed back to tell the cell body to stop firing. So it kind of self-regulates. If you take neuroscience with me, we'll talk more about that. But officially, most neurons only have one axon. There's different kinds of neurons, which we'll talk about neuroscience, but the main kind of neuron we see in the body only has one axon. Axons, not all of them, but most of them are covered in myelin. It's this fatty substance, and it insulates the wire, so your sodium ions aren't just leaking in and out. It makes the signal go faster, and basically the signal is going to go hop from the axon hillock to here, to here, to here, rather than going there. That takes longer. But when hop, I pushed a button. We can hop from node to node to node. That carries the signals faster. So cells that you want a fast signal tend to be myelinated. Myelin, That's that protein lipid that covers the axon. And as I said, it makes it go faster and insulate it, and it prevents the current from leaking out, the sodium from leaking out. MS, multiple sclerosis, is a disease of the oligodendrocytes, basically. That's the cells that myelinate in the central nervous system. And so your nerves just don't fire as well, and it can lead to all sorts of widespread damage, which we'll talk about in neuroscience, if you choose to take it. I'll close it out with just a few more things. Two types of cells that myelinate. One are called Schwann cells. Those are in the peripheral nervous system. And that's peripheral nervous system is all the nerves that go to and from your brain and spinal cord. And each Schwann cell, each individual Schwann cell wraps itself around a part, just one part of the axon. And so one axon might have, here's an axon, might have Schwann cell here, and here, and here, and here. Because each Schwann cell is going to wrap itself around the axon. It's going to insulate it and stop the sodium from leaking out there. Now, oligodendrocytes, those are a little different. One oligodendrocyte can myelinate up to 60 axons. That's going to be in the central nervous system, your brain and spinal cord. So it has little arms. So this arm can myelinate right here. This arm myelinates this patch right here. So one oligodendrocyte can make more bunches of myelin, and that's going to happen in the central nervous system. The node of Ranvier, that's the naked bit of the axon. That's where there is no myelin. And that's where we have a lot of sodium channels. And what an action potential is just sodium leaking in and potassium then leaking out. And so you don't want to have all these layers because that would get in the way of sodium moving. So it's the naked bit of axon. So there's a whole bunch of channels through which sodium can leak in. Saltatory conduction, saltare is Latin for to jump. And that's describes what I was showing before, how the signal hops from node to node to node to node. It doesn't bother trying to go through the myelin. It just hops from node to node. It's called saltatory conduction. And finally, the synapse. You need to know this word. What the synapse is, it's the space between one neuron and the next. So if this is an axon, and in red here's the axon terminal, and here's our next neuron. This is the soma and the dendrites of the next neuron. We call this first neuron the presynaptic neuron. The space in between them, here's a close-up of it, I'll get to that, is the synapse. And the second neuron is called the postsynaptic neuron. Now, in the real world, there's going to be thousands of neurons affecting this dude. And this one can affect hundreds to thousands as well. But we draw it one-to-one because, life is short. We don't have that much time. So many neurons can affect this one guy. And it's going to take in all that information and adjust what to do with it. So the space, this is a close-up here of the axon terminal right here. And here's a close-up of the dendrite on the next guy. The space in between is called the synapse. This first cell, so here in red, this is the presynaptic neuron. This is the axon terminal. Inside the axon terminal, there's going to be little vesicles. These are little balloons full of molecules of neurotransmitter. We'll talk more about this, I promise. So the presynaptic neuron, this is the axon terminal, has a whole bunch of little balloons, little water balloons full of neurotransmitter. And the postsynaptic neuron is going to have receptors. Those are going to be proteins of a specific size and shape that the neurotransmitter binds to. And with that, I will close this one out. In the next one, we'll talk about membrane potentials.